Okay, continue just now. 9 plus 2 microtubule arrangement. So 9 plus 2. Okay. Okay, this slide more about the tail. The 2 feet of each dynein along one doublet will alternately grip and release the adjacent doublet. Okay. The next scene will restrain the adjacent doublet from slide far. Okay. And then the combination of dynein and next scene, synchronized cycles make the movement of the tail. Okay, I hope you can imagine this one. You can imagine this one. Okay. So next, okay, cytoplasmic bridge. Remember, the another keyword of structure is cytoplasmic bridge. This cytoplasmic bridge is actually this one, this one, this one, this one. Okay, this one, cytoplasmic bridge. Throughout spermatogenesis process, from spermatogonia undergo mitotic division, and then undergo mitotic division 1 and 2, the sperm produced at each process is actually not separated between each other. They are attached together by this cytoplasmic bridge until they undergo spermiogenesis, when the round spermatid become elongate spermatozoa and then phagocytize of the cytoplasm. And then eventually the end of spermiogenesis, this cytoplasmic bridge will destroyed or degenerate. Okay, until sperm mature is complete, incomplete cytoplasmic division. So the cytoplasmic bridge function, it permits the cytoplasmic exchange among the 4 developing sperm. So from spermatid 1 until the number 4, okay, spermatid. So they exchange together the mRNA or whatever organelles are present. They distribute among each other. Okay. At the end of spermiogenesis, this cytoplasmic bridge rupture. What is the importance of this cytoplasmic bridge? Okay, remember during mitotic division 1 and 2, chromosome homologue separated, and then chromatid separated. Chromosome homologue separated. So we have 46 chromosome, and this 46 chromosome is actually paired up, can be paired up. So to make it more simple to observe, let's say the sex chromosome X and Y. So that is the chromosome homologue pairing up. Okay, mitotic division 1, this X and Y will, if these if these germ cells have XY, okay, so this chromosome homologue X and Y, completion of mitotic division 1, it will separate the X and the Y. Okay. So every process of spermatogenesis until spermiogenesis, so the spermatic spermatid that contain X and Y is different among these 4 spermatids. Okay, among these 4. So this diagram here 8. Completion of the division 2. Okay, this 4. So maybe this one X, this one Y, this one X, etc. So these 4 spermatids, and then the spermatids over here, okay, they exchange the cytoplasmic materials among each other. Okay, the X chromosome contains genes that code for cellular product essential for development of the sperm. Okay, so one of the spermatid having X chromosome, so during spermatogenesis and spermiogenesis, this X chromosome will translate, produce all the essential products for development, cellular development later after fertilization. So the spermatid that contain X chromosome only can produce that essential product. Once the X chromosome, the spermatid with the X chromosome produce that essential product, it will distribute to its friend, to the attached, the rest of the spermatids. Okay. Let's say if the cytoplasmic bridge rupture during this process, okay, between here, rupture here. Okay, so spermatid that have the Y chromosome, the Y bearing sperm will not able to develop and survive because rupture here lah. So because the X cannot distribute the the essential product for development to the Y chromosome. Okay, so that is the one of the special or important or significant of having cytoplasmic bridge. Alright. Okay, by the end of the spermiogenesis, when the spermatozoa form, the cytoplasmic bridge will rupture. Okay. So the next slide over here is about sperm morphology. The sperm morphology is elongated, the round shape. The sperm elongated, having the head, the midpiece, and also the tail. So the front view, we can see this oval shape here. Okay, so the side view, actually the oval shape at the side view is like a pointed shape. Alright, sperms. Okay, information about the sperm. Okay, sperm morphology. Different species will have different morphology, different shape of sperm. Okay, you can see the rat is hook shape, bull is more round shape, rabbit more smaller. Okay, different species have different shape of sperm. That's the point. Okay, after spermiogenesis, the spermatozoa will undergo spermation. Spermation takes place, which remove the remaining unnecessary cytoplasm and organelles. So it starts directly after spermiogenesis finish. Like it's like continue the cytoplasm being discarded. So this one remove the remaining unnecessary cytoplasm. So what is the definition of spermation? Spermation is the release of spermatozoa from the Sertoli cell. So the attached spermatozoa to the Sertoli cell now will be released from Sertoli cells. Okay. Okay. This is one type of maturation of the spermatozoa. So this spermatozoa, once released from the Sertoli cell, released into the lumen. So eventually it will release to the lumen travel to reach testes, and then go to the next one, vas efferentia, and then stored in epididymis. But when it travel to the reach testes to vas efferentia, this spermatozoa is lack of motility. They are being washed by the seminal tubular fluid. Okay, by the tubular, sorry, not seminal, the seminiferous tubular fluid washed the spermatozoa to reach testes. During this travel, the journey being washed to the reach testes, some changes happen also. Iron and small molecules changes. Okay, and then travel to vas efferentia. At that region, the spermatozoa will absorb. That region will absorb 90% of the fluid carrying spermatozoa. So spermatozoa is now more, what we call it, liquid, more condensed, right? Like the fluidity of the seminiferous tubule at vas efferentia, it's more condensed. And then next, the non-motile spermatozoa transported to epididymis. Here, the testicular fluid being more concentrated. Okay. So the movement of the spermatozoa is through, or the spermatozoa is through peristaltic contraction, because the fluidity over there is so concentrated. At epididymis, during the storage, the spermatozoa will undergo some changes. It will have more additional secretory product, glycoprotein, okay, like this slide over here. Okay, completion of sperm modeling, the nuclear condensation, this one is during spermiogenesis. Metabolism, cholesterol and phospholipid selectively metabolized and shifting lipid balance to the lipid balance towards diacylglycerol, meaning inside the spermatozoa, the lipid structure is in this diacylglycerol, unsaturated fatty acids, and demosterol. And then increased dependence of external fructose for glycolytic energy production. In terms of mobility, increase in disulfide linkage between proteins in outer dense fibers of the tail, so it will yielding a more rigid flagellum. Okay, CMP content in the tail also increase. So all of this is to help in terms of the sperm mobility after this. Okay, membrane being coated by glycoprotein, the membrane, the outer layer, the membrane of the spermatozoa, the sperms, coated by glycoprotein, and then rise in surface charge. This one due to sialic acid increment. So it will help in the next stage later on. Okay. So epididymis will add secretory products and glycoprotein, etc., as this table 9.1 during the spermation. So the spermatozoa attached to Sertoli cell being released from the Sertoli cell. During that stage, the residual body of cytoplasm being removed. At the same time, the spermatozoa being packaged, being packaged with the secretory products or other products during the journey, being washed from the testes to vasa efferentia and then stored in epididymis. Okay, and then the next process is capacitation. Capacitation is attainment of full fertilizing capacity. So to get this full fertilizing capacity, or the sperm get the capability of fertilizing the oocyte, so it will remove the coat and allow the sperm to bind to the egg. Remember the outer membrane we have coated with glycoprotein, right? And then all the surface charge. So during capacitation, this glycoprotein being removed, and then the charges also contribute to the attainment of fertilizing capacity. Okay, where sperm capacitation happens? So you can see here, fresh ejaculated sperm are unable or poorly able to fertilize. Okay, they must first undergo series of changes. These changes is actually the capacitation. So it's actually after ejaculation. So sperm capacitation occur now is in the female reproductive tract after ejaculation. Okay, capacitation associated with removal of adherent seminal plasma protein. Reorganization of plasma membrane lipids and the protein also seems to involve an influx of extracellular calcium and increase of CAMP and decrease in intracellular pH. Adherent and seminal plasma protein. Okay, refresh back. During spermiation, the spermatozoa travel or being washed from rib testes to vasa efferentia and then stored in epididymis. After they stored in epididymis, they being moved by peristaltic movement. Okay. Exit out from epididymis. So this seminal the sperms will exit out the epididymis, will go to the next structure, ejaculation duct, right? Before they enter the ejaculation duct, they will pass through the accessory organs. So accessory organs, seminal vesicle, prostate gland, bubourethral gland. So during their journey to that accessory organ, they will face some changes also. Okay, adherent seminal plasma protein. So meaning plasma protein comes from the seminal vesicle. Plasma membrane lipid and protein, some of it comes from the accessory gland. Okay, what happened during capacitation? So you can see here epididymal structure. Epididymis. The plasma membrane of epididymal spermatozoa contain a complement of surface molecules. Okay, which contain protein and carbohydrate, glycoprotein just now. Okay, and then undergo ejaculation duct, the accessory organ. Okay, pass through the seminal vesicle. Okay, the surface molecules of the epidermal sperm become coated with the seminal plasma protein. Okay, mask the portion of the membrane molecules. You can see orange in color. So this one yellow in color. And then once enter the female tract, these sperms will capacitated. Because exposure to the female environment tract, okay, the seminal plasma coating, the orange color, will remove, okay, along with some of the surface molecules. So exposing portion of the molecules, this one, okay, that can bind to the egg structure, zona pellucida. Okay, so recap of this sperm formation. Originate from primordial germ cell, PGCs. So PGCs will undergo spermatogenesis. Spermatogenesis, in spermatogenesis, mitotic division, and then undergo meiotic division 1, and then undergo meiotic division 2, producing early spermatid. And then this early spermatid will undergo spermiogenesis. Spermiogenesis is actually maturation of the sperm or transform the spermatid into spermatozoa. So during spermiogenesis, from round spermatid become elongated spermatozoa. By the end of spermiogenesis, we produce the elongated spermatozoa, removal of residual cytoplasm. So that is the starting point of spermation. So remove all the excess cytoplasm, and then the head of the spermatozoa detach or separate from the Sertoli cell. That is spermation. Once detached from the Sertoli cell, it will wash through reed testes, okay, and then wash through vasa efferentia, and then eventually stored in epididymis. From reed testes to vasa efferentia to epididymis, the tubular fluid, the seminiferous tubule fluid become more concentrated until epididymis. So the movement of the spermatozoa being washed is actually by the fluid current, okay, and then more concentrated to epididymis. Towards the epididymis, the movement is peristaltic movement, okay. During that movement, some changes happen also. Okay, glycoprotein being packaged, surface membrane, okay, and then they undergo to the next structure, the ejaculation duct. It will pass through the accessory organ, accessory gland. The accessory gland will again pack the sperms with all macromolecules needed, okay. Once ejaculated, entering the female tract, now because due to exposure to female tract, these sperms will undergo capacitation. So removal of the outer membrane, outer layer of the membrane, the sperm membrane. So it will expose the binding portion, the binding structure of the sperm. That's when the sperm able to fertilize the egg. Okay, so that's the end of the process from spermatogenesis until capacitation. After this, we look into hormonal regulation.